Dear learner, welcome you all in the course of Immunology BBYET 141. It is the elective courses for BSc General program. Dear learners, my name is Dr. Siaram. I am an associate professor, Discipline of Life Science, School of Sciences, IGNOU. Dear learner, I want to discuss today an overview of immune system. It is a very important topic. What is expected learning outcome of this session? Dear learner, uh, after studying of this session, after studying of this unit, you will be able to explain an overview of immune systems and discuss how body differentiate between self and foreign molecules. And after completion of this session, you will be able to explain the fundamental cause of the autoimmunity. And you will be able to list the postulate of the clonal selection theory. And you will be able to explain basic terms like pathogenicity and transmission. It is the expected learning outcome of this session. My presentation is divided into two parts. Part first, part second. In part first, I will discuss. In part first, I will discuss the historical perspective of immunology and classification of the immunity. But in case of the part second, I will discuss in detail how immune response will take place. What is the autoimmunity, self and non-self recognitions of our immune systems, and other thing, and clonal selection theory we will discuss in part second. In this session, I want to discuss what is the immune system. Immune system protect our body from invasion by pathogens. Our pathogens, if the pathogen they infect our body, the immune system will protect. And disease like allergy, autoimmune disease and cancer can result if immune system fail to work properly. If your immune system fail to work properly, several disease will occur like autoimmunity, cancer and infection will take place. Allergy will also take place if your immune system will fail, not working properly. And this can be distinguished. The immune system can distinguish between self and non-self molecules. And in immune system, cells that bind to self antigen are selectively eliminated during the early developmental stage. Only activated cells that bind to the foreign antigens are clonally selected and propagated. An immune system must combat the pathogens and prevent the infections. So our immune system is very strong. They will protect from the infection or they will prevent from the infection. It is a very important things related to the immune systems. What is the major activities of the immune system? There are the two major activities of our immune system. First one is the recognitions and second is the response. The immune system can differentiate even single amino acids among pathogen self and non-self molecule. So you can understand it is so strong, their recognition is so strong. If any single amino acid differences will be there, our immune system they will recognize it. After recognition of the pathogen, an effector response is modulated to neutralize the pathogen or toxin. So your pathogen will be neutralized, they will be eliminated from our body, or even the toxin if present, the toxin will eliminate. In subsequent exposure of the same pathogen generate a memory response. So it is a major activity of our immune system, recognition and responses. These are the two uh, major activities of the immune systems. Uh, let's discuss the historical perspective of immunology. These are the several scientists. They have done wonderful work. I want to discuss one by one. The immunity, first of all, it need to discuss what is the immunity. Immunity come from the Latin words immunis, which means to be exempted from the disease. And uh, Theodosius in 430 BC, while describing a plague in Athens, introduced the concept immunity. So it is first time 430 BC he gave the term the immunity. And another very important work is given by Lady Mary Wortley Montagu. In 1718, she observed the advantage of the variolations of the local population and got her children varulated. Their work is very remarkable. In this technique, dried crust from the pus of a smallpox patient has been given subjects by a minor cut or inhalation. So, with the help of the minor cut or inhalations, and the some dried crust of the pus of smallpox patient will be given, or ultimately the particular person they get virulated. Uh, the bold experiment led to a smallpox vaccine 75 year before the Jenners. Another very important scientist in this area, that is known as the Edward Jenner. He is an English physician in 1789, bent on improve the variolation techniques. He was surprised. He noted that milkmaid who has previously been infected with the cowpox were subsequently found to be immune to the smallpox. That means cowpox are able to eliminate the smallpox. He first time he observed, Edward Jenner observed, and Edward Jenner conclude that to the inoculating fluid from the cowpox pustule into subject might protect from them from the smallpox. So you can see, look at this figure. In this figure, you can see the Edward Jenner. They can give the vaccine to the particular children. Uh, Another scientist in this area is Louis Pasteur. So in 1879, the Pasteur worked with the disease chicken cholera, which were rampant in chicken coops at the times. So you can see in this experiment, here is the aged culture of the Pasteurella uh, maltosida. These are the aged culture of this bacteria is injected into the normal chicken, and no disease or death of this chicken are there. And in another group of the experiment, a fresh culture of the Pasteurella multicida is injected into the normal as well as immunized chicken, and no disease are there take place, but in case of normal the chicken will dead. So it is a this experiment gave the clue of the immunization. He first gave this is gave by the Pasteur, Louis Pasteur. So when Pasteurella multicida is inoculated into chicken, the chicken kill. And this principle of the vaccination was attenuated pathogen was discovered. Means there are some pathogen they are attenuated, and attenuated means nothing, it is a weakened their virulency. Virulency will be weakened of the particular pathogen, that is known as the attenuated pathogens. Means their pathogenicity will be reduced. So it is a very famous experiment are there. And another very important scientist in this area, that is known as the Emil von Behring in Sibas, uh, Sibasaburo Kitosato in 1890. Behring showed that immunity against diphtheria could be transferred from immunized patient by a serum to non-immunized individual. So he gave the very important concept of the immunizations, or it is due to the patient by a serum. And finally, the precipitin, antitoxin, and agglutination term came up. And they were considered the separate component. Even Evin Cabot in 1937 attributed all these functions. After some times, these immunoglobulins are now known as antibodies. Their experiment is very famous experiment, or ultimately they will discuss after some times this molecule they will recognize as immunoglobulins. And here is a very important scientist that is known as the Eli Mechnikov. He observed that WBC are able to phagocytize pathogen and other foreign material. It is a more active cells. In this cell, monocyte and neutrophils which are involved, or they are able to phagocytize the microbes in immunized animals. For a long time, the scientists were divided between the humoral and the cell mediated immunity. After some times, this type of the cells, it is divided into the cell mediated immunity and humoral immunity, and finally it was established by the both work together in the body. And another very important scientist in this area, Merrill Chase, in 1942, he successfully transferred immunity against pathogen causing tuberculosis while performing a leukocyte transfer experiment on the pig. And the lymphocyte was identified in 1950, an essential element of humoral and cell mediated immunity. And another very important scientist in this area, and Bruce Gillick, in 1952, he demonstrated by his experiment on the chicken that cell mediated immunity was affected by T cell and humoral immunity by B cell. So, here is the role of the T cell and B cell, which are very important in our body. T cell T cell is due to the cell mediated immunity, and humoral immunity, it is governed by the B cells. So, it is a role of the B cell and T cells are there. Another thing I want to discuss the classification of the immune systems. Our immune system is divided into the two main group. One is the innate immunity, and second is the adaptive or acquired immunity. So, innate immunity, basically non-specific immediate onset response will be there. But in case of adaptive immunity, it is very specific delay onset. After some week or some days, your adaptive immunity will fight against the particular pathogens. So, there are the two immunity are there. And adaptive immunity is further divided into the humoral immunity and cell mediated immunity. So, in case of humoral immunity, antibody will be produced. It is governed by the B cell. Another is cell mediated immunity. It is governed by the T cell activations. So, after T cell activation, or ultimately the pathogen will killed. So, here is the interaction between the two type of immunity will take place, or ultimately we will protect from the pathogen. The interaction between the two immune system is very important. Ultimately we will fight against the pathogens. Innate immunity. So, in this slide I want to discuss the innate immunity, innate immune systems. The innate immune system is a first line of defense of the body that is present from the time of the birth. Basically, we will acquire this immunity at the time of the birth. The innate immunity component are present before the initiation of the infection. So it is a very important innate immunity component are present before the initiation of the infections. And innate immune system is not specific for a particular pathogen. So it is not specific for the particular pathogen. If any pathogen will come, it will respond. In general, increase the ability of the body to recognize commonly found molecules on the surface of the microorganism. So basically, the microorganism and their surface, they have the different type of the molecules are there, or ultimately they will provoke the immune response in our body. In this case, I want to discuss the various barrier of the immunity which come under the humoral immunity. The first one is the anatomical anatomical barriers. The skin has a epidermis and dermis that form to covering and prevent pathogen invasions. The low pH of secretion of the sebaceous or sweat glands in inhibitory to the pathogens, means the low pH which are available in our stomach and sweat gland, they will produce the inhibitory substances against the pathogen. So ultimately pathogen will reduce. The mucous membrane from a continuous lining where the pathogen are trapped in a mucus. The mucus is another anatomical barriers are there. Microorganisms compete with the normal microflora of the body for attachment site. So these are the, in this figure you can see it, these are the various anatomical barrier are there. Here is the liver, skins, and mucous membranes. The various things are there, or ultimately they will protect from the pathogens. Another is the physiological barrier. The physiological barriers, the temperature, mainly temperature, pH, and antimicrobial substance, it come under the physiological barrier. Temperature, fever protect against the pathogens, and high body temperature of some organism protect from the disease. For example, chicken are protected from the Bacillus anthracis due to their high body temperature. So in case of the chicken, the body temperature is very high, so the Bacillus anthracis are not able to infect to the chickens. And low pH, pH 2.9 of the gastric secretion inhibits the most of the microbes. And in case of the stomach, it releases the pH is 2.5, and the gastric secretion they contain the 2.5 pH. pH is very less, so ultimately the microorganism will kill. Antimicrobial substances like lysozyme present in tears, saliva, and they are able to protect against the pathogens. So it is a physiological barriers. And third one is the phagocytic barrier. The phagocytic cell engulf, break down and kill the pathogen. Example are the macrophages, neutrophils, and monocytes. So these are the three main important cells which are involved in the phagocytosis. The phagocytosis, they are able to phagocytize the bacterial pathogens, or ultimately they will kill the pathogens. And fourth one is inflammation. It is very important barrier are there. There is a fluid leakage from the blood vessels due to the vasodilation and increase the permeability, and there is a influx of the cell, the area of the pathogen invasion or damage. So there are the four type of barriers are there, or ultimately they are able to protect from the body. And it is the adoptive and acquired immunity. So I have already discussed it, humoral immunity, and it is the acquired or the adoptive immunity. So in the it is divided into two parts. I have already discussed it. First one is the humoral immunity. The humoral immunity, uh, it will govern antigen presentations. Basically, it will work with the antigen presentation. The formation of viral protein 5 macrophages. The several viral protein, they will present on the surface of the macrophages, and the formation of antigen MHC complexes. So ultimately humoral immunity will take place, or it will kill the pathogen. And T cell activation, so the T helper cell will activate in case of the humoral immunity, or ultimately immune response will take place. The binding of the T helper cell to MSC antigen complex, it is one process. Ultimately your T cell will be activated. And second is the release of the cytotoxin from the macrophages. So your macrophages, they will produce the cytokines. It is a very important chemicals which are released by the macrophage, or ultimately they will involve, they will provoke the immune response, or ultimately kill the pathogen. And activation of the T helper cell to release the cytokines. And B cell activations, here it is governed by the B cell. I have already discussed it. And activation of the virgin or memory cell by cytokines, and promotion of the mitosis and differentiation of the B cell. And B cell, and fourth work in the humoral immunity, the production of the antibody, and releasing and circulating of antibody into the blood stream from the plasma cell. So these are the humoral immunity are there. They in this case, basically B cell, they will release the antibody, or antibody, they are able to neutralize the pathogens, and it will able to remove the pathogens from our body. And another is the adoptive or acquired immunity. In case of adoptive and acquired immunity, or you can say it is the cell mediated immunity. Um, in this case, there are the same things which are in the humoral immunity, antigen presentation and T helper cell activation, it is same, but it is a different. There are the two things which are different in case of the cell mediated immunity. One is the T cell activations. In this case, activation or version of the memory cell by the cytokines. So cytokines, they will help to the activation of the T cells. And another is the promotion and mitosis or differentiation of the T cytotoxic T cells. And ultimately this cellular destruction, the binding of the effector T cells to the infected body cells, and induction of the cell lysis by releasing of the perforins. The perforins is to the group of the molecules, they can lyse your pathogens. So these are the two type of the immunity are there. One is the humoral immunity, another is the cell mediated immunity, or they will protect from the infections. Dear learner, I want to summarize part first. You have learned the number of the things, like immune system is a defense system to protect animal against the pathogen and cancers. And you have also learned the body has innate and adaptive immunity systems, or they will work together, or ultimately we will get rid of from the infections. Innate immunity is a first non-specific line of defense of the body present from the time of the birth. And the adaptive immunity system further has humoral and cell mediated immunity. And self and non-self recognition are essential feature of the immune system. The dear learner, we will take a short break. We will don't go away. We will come within 2 to 3 minutes.
Are you interested in television journalism or radio journalism or online journalism? Or do you want to learn with the flexibility of open learning? Or if you are a working professional wants to enhance your knowledge and skill with certification in the field of electronic media, then School of Journalism and New Media Study IGNOU offers PG Diploma in Electronic Media for you. This program includes audio and audio-visual program production for radio, television and online platforms. Here are some important details about the program. We have total 8 courses of 32 credits, 6 theory courses, 1 practical and 1 project. Eligibility: bachelor's degree in any subject. Minimum duration 1 year, maximum 3 years. Medium is English, but students can write their examinations, assignments, practical and project in Hindi also. So if you are interested in television, radio or online media, please come and join IGNOU's PG Diploma in Electronic Media.
Dear learners, welcome in my next part second. In this part, we will discuss various things like cell involved in the regulation of the immune response, autoimmunity, self and non-self recognitions, clonal selection theory, and at last we will discuss how antibody involve in destruction of the pathogens. So I want to discuss one by one in part second. First of all, how cell involved in the regulation of the immune system. So there are the 3 type of the cells which are involved in the immune response. First one is the B cell, second is the T cell, and third one is the antigen presenting cells. In case of the B cell, form and mature in the bone marrow. So their maturation and their formation it will take in the bone marrow. Bone marrow is a very important part of our body. They will mature our B cells. They express membrane bound antibody molecule with specificity to a particular antigens. And when the naive B cell encounter an antigens, it is activated and proliferate to form memory B cell and plasma cell. So it is a very important function of our B cell. If it activate, proliferate and it form the memory cell and plasma cell. The plasma cell they are able to secrete the antibody very rapidly. And memory cell get activated on subsequent exposure to the same antigen and produce a much more severe response against the particular pathogens. The B cell which are very important in our body, they will protect it, it will govern our humoral immunity. Another is T cell, it is formed in bone marrow but mature in the thymus and express T cell receptor on its surface. So you can see this figure. Here is the cells, T cells are there they have the TCR, T cell receptor are there. They are able to recognize the particular molecules. The T cell can recognize antigen when they bound to the MSC molecule. The helper T cell on activation secrete cytokine and further activate cytokine T cell. So your cytokines, which are they have the very important role. Your T helper cell, they will activate or ultimately they will secrete the cytokines and further it activate the cytotoxic T cell, and T cell proliferate to form the cytotoxic T lymphocyte that kill the pathogen. So it is a function of the T cells, or ultimately we will protected from the pathogens. And third one is the antigen presenting cells. In antigen presenting cell, there are the three type of the cells are there: macrophage, dendritic cell, and B lymphocytes. And it is antigen presenting cells and express class second MSC molecule on their surface. So basically class second MSC molecule which will express on the surface of the antigen presenting cells like macrophage, dendritic cells, and lymphocytes. An antigen presenting cell internalize and internalize the antigens and they degrade it into a smaller fragment, and this fragment and smaller and the fragment is then expressed with the MSC second molecules on the surface, and they provide the co-stimulation signal for the T helper cell activation. After T cell helper activation, it will activate C cytotoxic cell, or ultimately they will kill the pathogens. Next move to the next topic, antigen processing. How antigen processing take place? The T cell recognize antigen only after degrade into smaller peptides that can form the complexes with MSC class first and MSC class second, and this process, that is antigen undergo a called antigen presentation or antigen processing. So basically in this case, if any pathogen they infect our body, the macrophages and dendritic cells, they are able to process it, antigen processing, and presentation will take place. And the route of antigen entry determine whether antigen will be presented with the class first MSC molecule or class second MSC molecule. The antigen presenting cell are the two types. One is the exogenous antigen processing, another is endogenous antigen processing. In case of exogenous antigen processing, it take place, it taken up by the cell through the phagocytosis or endocytosis. So first of all, if pathogen infect our body, it will taken up by cell through the phagocytosis or endocytosis and degrade into smaller peptides. So your pathogen, your viruses, they infect our cell, it will degraded into smaller peptides. APC perform the processing like macrophage, dendritic cell, or B cell. B cell, in case of B cell, endocytic processing will take place. So it will perform antigen presenting cell processing will take place. The antigenic peptide produce are bound to the MSC class first molecule and transported to the cell in case of the exogenous antigen presentation. So the presentation of the exogenous peptide is limited to the APCs and that bear MSC second molecules. Now the CD4 displaying T cell bind with MSC second bound antigen. These cells act as a helper T cells. So ultimately they will activate your T cells, or it will kill the pathogens. Another is the endogenous antigen processing. In case of endogenous antigen processing, the antigen are produced inside to the host. So by name you can understand it is the endogenous antigen processing. The common sources of endogenous antigen are cancerous cell and virus infected cell that are continuously from the virus proteins, forms viral protein. They are degraded into endoplasmic reticulum into a small fragment. These fragments bind to the MSC class first. So you can see it in case of the endogenous antigen, the MSC second molecule will involve. The MSC second molecule moves to the cell surface. The class first MSC antigen complex is recognized CD8 bearing T cell and cytotoxic cell kill these cells. So these are the function of the endogenous antigen processing and endogenous antigen processing. Another thing I want to discuss, self and non-self antigens. So what is the self and non-self antigen? Our immune system should be able to distinguish between the own cell or foreign invader. Foreign invader. The foreign invader, it may be virus, it may be bacteria, it may be fungi, helminthes, and various pathogens are there. But in case of own cell, a number of the tissues are there, a lot of cells which are present inside to the body. So our immune system, they are able to recognize it, the self and non-self antigens. And pathogen have antigens. Basically, these antigens present on the surface of the microorganisms or pathogens that are not found into the human, thus and thus are recognized as a foreign. So your pathogens, they will recognize as a foreign materials. MSC is the most important. So in case of cell and non-self recognition, MSC is another very important self identification molecules. Each person has unique combination of the MSC molecule that are recognized as their own. Is the activated and attack these cells and they differences when any differences is detected in these molecules. If any foreign invader or the self antigens are own cells like infected cell, cancerous cell and transplanted tissues, then your immune system will be activated or ultimately it will kill it. And B cell antibodies, B cell have antibodies attached to their surface that recognize and bind to the specific antigen or pathogen surface. And T cell activation occur in the antigen presenting cell like macrophage and dendritic cell. So these are the several things they will make our system to recognize self and non-self. Means B cell which are involved, T cell which are involved in the pathogen that internalize the antigen presenting cell, broken down and fragment expressed with MSC molecule present on the surface. Another cells which are involved or ultimately your self and non-self system will make perfect with the help of the antigen presenting cells. And T cell bind with the presented fragment by the specific T cell receptor, and this lead to the T cell activation. So these are the various things which are they are able to make your systems, they are able to recognize self and non-self. Otherwise your immune system, they will not recognize it. It will create a chaos inside to the body. Another thing I want to discuss, immunological tolerance. The ability of the T cell and B cell to ignore or tolerate self antigen is called the immune tolerance. Means immune tolerance against the particular cells, our T cell, our B cell, it is tolerated inside to the body. But any foreign material or pathogens or any foreign invader or material, they will intolerate against the immune system. Your immune system will provoke the immune response or ultimately they will kill it. So their immunological tolerance, it is very important phenomena inside to the body. The immunological tolerance involved to the selective elimination of self-reactive cell during the development. During the development, if self molecules are there, it will be eliminated during the development. And a T cell bind to the self antigens. Antigens are killed in the thymus and likewise B cell in the bone marrow. The tolerance gained by such mechanism is called the central tolerance. The central tolerance is a very important phenomena in our body. They will recognize, they will maintain the self and non-self system to the body. So immune system can develop cell with specificity and antigen only through the process of the tolerance and somatic recombination will take place. Ultimately they will maintain the immunological tolerance. It is a very important phenomena of our body. Another another thing I want to discuss, that is known as the autoimmunity. Autoimmunity losses loss or self tolerance refer to the immune response to develop against one's own cells and tissues. Means they will not recognize it as a self. So ultimately immune response will take place. The loss of the cell tolerance will be take place, that is known as the autoimmunity. It may be organ specific, localized or systematic. So it may be organ specific, or particular organs are there like liver, kidney or any particular organs. It may be so your immune response against the particular organ, or it may be localized, particular location, it may be possible, or it may be systematic. Genetic background play a very important role in the extent and severity of the disease, and sometimes it is very severe and sometimes it is not severe. So autoimmunity, virtually all immune response of the T cell dependent. So your autoimmunity basically it is T cell dependent and not the B cell dependent. The formation of the new or altered epitope, it may be another problems of the autoimmunity. It is divided into the four: sharing epitope between tissues and agent, or it may be molecular mimicry are there. Then autoimmunity will take place against the particular agent or particular tissues are there. Exposure of the hidden antigen, it may be another cause of the autoimmunity. In viral infection, it may be cause of the another autoimmunity. Lost the control of the lymphocyte response. So autoimmunity which are involved, which they can create if your self tolerance will loss. Another thing I want to discuss, the clonal selection theory. So how the cell, they select the particular cells or ultimately they will produce the antibodies. So your clonal selection theory, it is a most accepted theory worldwide. It is proposed by the Niels Jernnes and Frank McFarlane Burnett in 1957. And Niels Jernnes proposed that large amount of antigen specific antibodies are antigen specific antibodies are always present in the serum. So he gave the uh, idea the that the large amount of antigen specific antibody are always present in the serum. And Burnett explained Jernnes theories and gave the clonal selection theory. It is a widely accepted theory. In clonal selection theory explain the origin of antibody formations. The clonal selection theory states that each immunological component B cell has the receptor that can combine with one antigens. After binding of the antigen, B cell is activated, causing it to proliferate to form the clones of the cells. Uh, you can see in this figure. You can see in this figure, it is a antigens are it is a cells are there, they have the antibodies, they can recognize it or it will proliferate. You can see the number of the cells are there. If again antigen will come and it will clone will forms, and or you can see it again it differentiate into the plasma cell and memory cell. So it is due to the clonal selection. So your clone, the particular clone will select against the particular pathogens or particular antigens are there. These are the antigens are there, they can recognize it, it will destroy it, and your cell will generate. If again any antigen will come, they will bound it or it will proliferate. You will get the plasma cell and memory cell. Plasma cell, they are able to generate the antibodies. Then antigen select to the single clone from the proof of the B cell clone and various specificity during the antibody production. Antigen select B cell differentiate into plasma cell and antigen specific memory cell after the antigen selected proliferations. In each, it is a postulate, and clonal selection theory postulate. So basically, this postulate given after the theory, that is known as the clonal selection theory. The postulate is made. Um, the each lymphocyte has a receptor that recognize a specific antigen. Means each lymphocyte has a receptor that recognize a specific antigen. The lymphocyte, they have the receptor, they are able to recognize a specific antigen. The cell activation occur only after interaction with the particular antigen. If antigen will be not will be there, the cell activation will not occur. After interaction with the particular antigen, then activation will occur. Otherwise, cell activation will not occur. The clone of the cell is created and has been same antigen specificity of its parent cells. The lymphocyte with the endogenous antigen and their receptor are destroyed selectively, and this slope of this stop the immune system from the attacking self. So it is a clonal selection theory postulate. Another thing I want to discuss the various various immune response generate against the viruses. Here is the virus, you can understand here is the viruses are there. It is a various immune response generate against the virus. So here is the IgA prevent viral binding host cell. So IgA is basically antibody are there, they will prevent the viral binding to the host cell. So ultimately your virus will not infect to our body. Another is IgA, IgM, IgG block the viral envelope fusion with the host plasma membrane. So ultimately your virus, they are not able to infect our cells, our body. IgG, IgM promote the opsonizations. So your opsonization will promote ultimately your macrophages, they engulf the particular viruses or particular pathogen or particular antigens are there. Another is the complement mediated lysis involve viruses by a membrane attacking complex that is known as the MAC. The MAC will forms against the particular pathogen or particular virus, or it will kill the pathogens. And antiviral activity by the interferon gamma. So after infection with the particular virus, it is a very important molecules are there that is known as the interferon gamma. Interferon gamma, it will release inside to the body, or it will reduce the infections of the infection, like the infection through the viruses. And CLT skill be the complete kill virus infected cells. It is another NK cell natural killer cell, macrophages kill the infected cell, antibody dependent cell mediated cytotoxicity, ADCC. So these are the mechanisms, various mechanisms which are involved, are they are able to generate the immune response against the pathogen. Ultimately your virus will kill, or virus will eliminate from the body. So these are the very important 6 mechanism are there, or it will provoke your immune response, or ultimately it will um, reduce the pathogen infections. Another thing I want to discuss, these are the different neutralization mechanism by the bacteria, bacteria by antibodies. So these are the very important compounds are there, antibody which are produced inside to the body. I have already discussed it. It is released by the B cell. The B cell release antibody, they will kill the pathogens. So you can understand here is the antibody bind to the pathogens. Here it is a pathogens. The antibody bind to the pathogens, and along with the C3b. C3b is a very important factors are there. They promote phagocytosis by the opsonization. So here is role of the C3b is very important. It is a very important factors are there. So after binding of this, so your pathogen, they will phagocytize. So here is role of antibody is very important. They will involve in the phagocytizations. Another is toxin neutralizations. So your antibody, they are able to neutralize the particular toxin which are released by the particular pathogens, like E. coli. E. coli, they will produce the particular toxin like cigatoxin and labile heat toxin and labile toxins. The various toxins, pathogenic E. coli, they will produce a lot of toxins are there inside to the body. It will neutralize by the particular antibodies. The antibody toxin complex form which is cleared by the phagocyte cells. And third one is the mast cell degranulations, and C3a and C4a, C5a act as a anaphylotoxins are the causes mast cell degranulation. Thus the extra vacations are extra vacations of the neutrophils and lymphocytes from the blood and tissues. So these are the very important mechanism which are involved, or ultimately they will neutralize the pathogens by the opsonization, by the phagocytosis, and by the anaphylotoxins and various mechanisms. Degranulations, the mechanisms are there. Ultimately it will form the granules of the pathogen, or ultimately the kill the pathogens are there. So these are the mechanisms which are controlled by the antibodies. Dear learner, I want to summarize my topic, this session, part second. You have learned the ability of the T and B cell to ignore the tolerate self antigen is called the immunological tolerance. So you have also learned the clonal selection theory, and the clonal selection theory, that each lymphocyte express antigen specific membrane receptors, which receptor specific against the particular antigens. And here is you have learned also the role of the T helper cell and cytotoxic T cell. And T helper cell and cytotoxic T cell are two subpopulation of the T lymphocytes. And T helper cell recognize antigen MSC second complex, which regulate the immune response by secreting the cytokines, and cytokines T cell display cytotoxic activity by recognize antigen first MSC molecule. So dear learner, if you want to read it, you can go through the immunology courses, BZYET 141 unit number 3. So you will find out details of your courses. you can you can go through, or ultimately you can learn the various things related to the this session. And thank you so much.